Hello everyone, and welcome to Lecture 6 of Unit 1. In this lecture, we will go over eukaryotic cell structure and function. Some of this is going to be a review. If you took introductory biology recently, it will probably be very familiar to you. For those of you who took that class a little while ago, this will help kind of jog your memory and remind you of some of the big concepts in terms of eukaryotes, cellular structure and function. And for everyone, what I want you to be doing when you listen to this lecture is thinking about how eukaryotes are similar and how they're different to prokaryotes. So one of the big objectives for the class is to help you understand the main differences between prokaryotes and eukaryotes in terms of their cell structure and function. As usual, here are your lesson objectives, and the way I tend to recommend using these is to look at them now before you get going in the lecture, make sure you know specifically what you're listening for, and then look at them again, give them another kind of review, check them over after you've read the chapter, listen to the lecture, and make sure that you have understood all of the key points. To start us off, I want to talk about the origin of eukaryotes. Most life on Earth, most types of living organisms, are actually prokaryotes. So remember, you've seen this diagram before. This is the diagram of the 3 domains of life on Earth, and 2 of the 3 domains of life on Earth are prokaryotes. So basically, about 2/3 of living organisms, or at least 2/3 of the types of living organisms, are prokaryotes. And for about 1 billion years or so, only prokaryotes existed on the planet. So when life first developed on planet Earth, the only life that existed was prokaryotic life. And it wasn't until about 1 billion, maybe 1.5 billion, 1.25 years after that, that we start to see evidence of the development of eukaryotes. So one of the big questions that scientists had about this is, what happened that all of a sudden led to this second type of cell on the planet? If for over 1 billion years, or just right around 1 billion years, all of the life on Earth was prokaryotic, what event happened, or what series of events happened to allow this other type of cell to develop? And after lots of different hypotheses and a lot of different testing and a lot of different thoughts about this subject, there arose one of the most plausible explanations for how we all of a sudden got eukaryotes, even though prior to that we just had prokaryotes. And that explanation is called the theory of endosymbiosis, or endosymbiotic theory. And what endosymbiotic theory basically states is that eukaryotes arose because of the combination of 2 different types of prokaryotic cells, or sometimes 3 different types of prokaryotic cells. So we're going to go to the next slide and talk a little bit more about how that happened. But basically, this slide just gives you an introduction to answering one of the big questions about life forms on Earth, and that is, how did we go from having just one cell type, the prokaryotes found in the domain bacteria and archaea, to having now 2 distinct types of cells on the planet, with that second cell type being the eukaryotes. So endosymbiosis itself is a process in which a large cell engulfs a smaller one and they live together. This process appears to have occurred several times throughout the history of life on Earth. And it was this process of a large cell engulfing a smaller one and them managing to live together successfully that appears to have led to the development of eukaryotes. So most of the time when a large cell engulfs a smaller one, the smaller one gets digested. It dies, and the bigger one uses it as a source of nutrients and then gets rid of anything that it doesn't need. However, there seem to have been some instances where larger cells engulfed smaller ones, and instead of the smaller one being digested, that arrangement that they had worked out well for them. So in this instance, what I'm showing here is you've got a big purple cell and a small green cell, and the purple cell engulfs the small green cell. And instead of the green cell dying and being digested and just being used for metabolic activities, in this situation, this arrangement worked out well. And basically, the small green cell got protection from the big purple one. Maybe it got given some extra nutrients that the purple one didn't need anymore or wasn't going to use. And the small one maybe took those nutrients and produced some extra energy that it didn't need anymore. So the purple one is able to use that extra energy. And this arrangement basically benefited both of them. Because this was a beneficial arrangement, it ended up being passed on through generation to generation. So here I've just got an example of one generation being created with this arrangement because the slide was getting too busy and too full if I tried to show multiple generations. But essentially what you can see is because this arrangement was beneficial, when this cell or this complex goes to replicate, everything gets passed on. So the next generation also has a small green cell in a big purple cell. And then each of these would go to replicate, and this arrangement would get passed on through the generations. At the point at which this arrangement had been passed on enough and the two individual cells could no longer be separated from each other without both of them dying, that was the point in time where we can say that is when we had the first eukaryotes. So it wasn't just when a big cell engulfed a small cell and it kind of worked out for a little bit. It was the point at which we could not separate these two cells anymore and have them survive any longer. That point where they become dependent upon each other, that is the point we can say that's when eukaryotes truly developed. When the two prokaryotes, or the two original cells that were prokaryotes, became dependent upon each other, that's when eukaryotes arose. Now, this might seem a little bit far-fetched, but we actually have quite a bit of evidence for it. And the strongest evidence we have lies in the organelles that we call mitochondria and chloroplasts. Mitochondria and chloroplasts are found in eukaryotic cells. Things like plants have both mitochondria and chloroplasts. Animals have just mitochondria. But basically, if you look at a mitochondria and chloroplast and you study it, what you will find is that one, they are the same size as prokaryotes, as other bacteria that you can find just in the environment today. Two, they have their own DNA. So in a eukaryotic cell, in the nucleus, you will have the DNA that belongs basically to that eukaryotic cell. And if you were to be able to pop open a mitochondria or a chloroplast, you would find that they actually have their own chromosome that's contained within that structure. That chromosome is circular, just the same way prokaryotic chromosomes are circular. And if you look at the sequence of the nucleotides, you look at the sequence of the A's, the G's, the C's, and the T's on that chromosome, you will find that it is an extremely similar sequence to bacteria that are living today out in the environment. They also have their own ribosomes. So again, if you were to look at a eukaryotic cell, you would find ribosomes in the cytoplasm and attached to the endoplasmic reticulum, and those ribosomes, of course, are going to help with protein synthesis. But if you were to pop open a mitochondria or a chloroplast, then you would find ribosomes in there that are similar to the prokaryotic ones. So as a reminder, the size of the prokaryotic ribosomes is 70S, and that actually happens to be a different size than eukaryotic ribosomes. We'll get to talking about the size of eukaryotic ribosomes on a future slide, but just for now, this is kind of the information you want to focus on this slide. Now that we know where eukaryotes came from, we're going to go ahead and start discussing the general characteristics of eukaryotic cells. Of course, because there are many types of eukaryotic cells, just like there are many types of prokaryotic cells, there are going to be differences in terms of the actual specifics that we find in the various types of eukaryotic cells that are out there. But we're just going to be going over the general components of eukaryotic cells. Now, one thing to keep in mind is that eukaryotic organisms can be single or multi-celled. Remember that prokaryotic organisms are always single-celled, but a eukaryotic organism can be made up of just one eukaryotic cell, or it can be made up of multiple eukaryotic cells that combine together and kind of work together to help that organism survive. And one of the big things about eukaryotes, especially when we compare them to prokaryotes, is that they are compartmentalized, which means they have a lot of compartments inside their cytoplasm. Those compartments are also called membrane-bound organelles, which means they are small structures surrounded by a membrane that are dedicated to performing a specific function or a specific task for that cell. Compartmentalization allows for a couple of things that prokaryotes can't do. One is that it allows for the cells themselves to be bigger. If you were to look at a eukaryotic cell and a prokaryotic cell right next to each other on the same slide under a microscope, you would find that almost all the time the eukaryote is bigger than the prokaryote. And part of the reason for that is because of the compartmentalization of eukaryotes. This compartmentalization also allows for organisms to become multi-celled. So while a cell is always just a cell, a single individual unit, remember we can have eukaryotes in terms of the eukaryotic organism as a whole that can have multiple cells in it. So being able to compartmentalize allows individual cells within a multi-celled organism to perform specific jobs rather than having every cell in that organism doing the exact same thing. So in a prokaryote, it is always only just one cell, and it has to do everything it needs to survive by itself. Every single metabolic reaction, every task, every moment of its entire life, it has to do alone. Whereas in a eukaryote, if you have multiple cells that are combined, some of the tasks can be given to certain cells and other tasks can be given to other cells, and they work together for a common purpose or a common goal. So just like with prokaryotes, we are going to go ahead and talk about the individual structures that we can find in a eukaryotic cell and talk about the functions of those different structures. And we're going to start from the outside and work our way in. Again, some people like to start from the inside, work their way out. That is fine too. It's all the same information. But for me, it's more logical to start from the outside and then work our way in. Eukaryotic microbial cells tend to have a glycocalyx, similar to the glycocalyx found in prokaryotes. The glycocalyx is composed of polysaccharides, lipids, proteins. So again, very similar to what we would find in the glycocalyx of prokaryotes. And for certain eukaryotic microbes, especially things like algae, they can, the glycocalyx can be influential in helping biofilms form. So in the environment especially, a lot of times we will find algae, which are oftentimes eukaryotic organisms, and they will join their glycocalyxes together, create that matrix, and start developing a biofilm. Underneath the glycocalyx, two types of eukaryotic microbes have a cell wall. So when we talk about the eukaryotic microbes, we are talking about fungi, algae, protozoa, and helminths, and two types of eukaryotic microbial cells will have a cell wall, the fungi and the algae. Now the composition of that cell wall is different than what we would find in prokaryotes. In fungi, that cell wall can actually be made up of a lot of different types of molecules. One of the most common types, and this is not the only one, but one of the most common types is cellulose, which is just a bunch of glucose joined together. Another type can be actually silicon dioxide, which is essentially glass. We'll talk more about those cell walls when we get to algae later in this lecture. In fungi, the cell wall is made of what we call chitin. So in fungi, there's only one type of molecule that makes up the cell wall, and it is called chitin. That's kind of interesting because the exoskeleton of insects is also made of chitin. So they have something in common. They share that molecule. Now next, we will find the cell membrane. So if we're talking about a fungus or an algae, the structure goes glycocalyx, cell wall, cell membrane. If we are talking about a protozoa or a helminth, we would be talking about the structure going glycocalyx to cell membrane. So keep that in mind. There's a slight difference just depending on which type of microbe you're talking about. But the cell membrane is a typical phospholipid bilayer. It is similar in terms of structure and function to the cell membrane we find in prokaryotes. It helps with various cellular reactions, plays various roles in metabolism, can help with transport in terms of getting things into the cell and getting things out of the cell. But while the plasma membranes of prokaryotes and eukaryotes have a lot in common, there is one notable difference. Eukaryotes contain a molecule called a sterol in their cell membranes. So sterols are a group of lipids, and there are a lot of different individual types of sterols out there. One common sterol that probably everyone has heard of is cholesterol. Cholesterol is the type of sterol that is found in the cell membranes of humans and other types of animals. But basically, sterols help provide kind of stability and flexibility to the eukaryotic cell membrane. Prokaryotes typically do not have sterols, and the few types of prokaryotes that do have sterols basically stole them from eukaryotes. So in general, if you find sterols in a membrane, that is a eukaryotic cell. Occasionally, we will find a prokaryote that has some sterols in it, but that's only because they stole them from a eukaryote. They don't make their own the way that eukaryotes do. We're going to move from the external part of the eukaryotic cell to the internal part. And on this slide, we're going to cover the major organelles. Of course, just like prokaryotes, there is cytoplasm, there's a cytoskeleton as well, but some of the other things that are very obvious when we look in a eukaryotic cell and that tend to take up a lot of space are the three following structures: the nucleus, the endoplasmic reticulum, and the Golgi apparatus. And these, along with a few other things, oftentimes are referred to as the endomembrane system or part of the endomembrane system. So the nucleus, of course, is kind of the major organelle of the eukaryotic cell. Typically, it's roughly central. It's not always centrally located, but roughly it's in the center of the cell. Within the nucleus, there is the DNA, which of course provides all of the instructions for making all of the proteins that the organism needs to make and is passed on to next generations. And the big differences between the DNA found in prokaryotes versus the DNA found in eukaryotes is that in prokaryotes there is a single circular chromosome, while in eukaryotes there are typically multiple chromosomes, and most of those chromosomes are going to be linear. So they're going to be straight lines. Another unique feature to eukaryotes when we compare them to prokaryotes is that prokaryotes have just one copy of every gene they need, but eukaryotes have two copies of every gene they need. So basically, prokaryotes, the one gene that they have that codes for a protein that performs a specific task, has to function perfectly all the time. Eukaryotes, though, have a backup, essentially. So they have two copies of every gene they would need, or basically two copies specifying every protein they would need to create. And those two copies allow them to kind of have a backup in certain situations if one of the copies of the genes is not so great. Now, next to the nucleus will be the endoplasmic reticulum. The endoplasmic reticulum is used to make macromolecules. There are two types of ER, or endoplasmic reticulum. The smooth, which is responsible for making carbohydrates and lipids. CHO is an abbreviation for carbohydrates, just to remind you of that. And then the rough endoplasmic reticulum is called that because of ribosomes that are attached to it. So when you look at it under the microscope, it looks like a little bit grainy, like there are little grains attached to it. But anyway, the rough endoplasmic reticulum is responsible for making proteins. Near the edge of the endoplasmic reticulum, we will find the Golgi apparatus. The Golgi apparatus essentially takes the macromolecules that were made in the endoplasmic reticulum and it kind of packages them up, helps finalize them, get them into their final form, and processes them so that they can be moved either internally, so somewhere else inside the cell, or externally. If the cell is creating a molecule that's going to be excreted from the cell, the Golgi apparatus can package that up and help get it to the plasma membrane so that that molecule can be sent outside the cell. Now, as promised previously, I told you we would talk a little bit more about eukaryotic ribosomes. Just like prokaryotic ribosomes, eukaryotic ribosomes are the site of protein synthesis. There is a big subunit and a small subunit, and there's a space in between those subunits where messenger RNA sits, tRNA or transfer RNA is brought in, and the amino acids that are on the transfer RNA will attach to each other through peptide bonds and form a polypeptide, which is the beginning phase of a protein. In eukaryotes, the big subunit is what we call 60S, the small subunit is 40S, and in everywhere else, 60 and 40 would add up to 100, but in microbiology, in this particular situation, it adds up to 80. So prokaryotes have a ribosome size of 70S, and eukaryotes have a ribosome size of 80S. This may not seem like that big of a deal, but it is really critical. And it's important for multiple reasons. One of the big ones in microbiology is that many antibiotics, which are chemicals that are used to deal with bacterial infections, work to stop 70S ribosomes from working, but not 80S ribosomes. So what that means is these antibiotics work to stop protein synthesis in prokaryotes, but not to stop it in eukaryotes. And that is critical because when a person or an animal or potentially even a plant has a bacterial infection, we want the antibiotics to work on that bacteria and interfere with that bacteria without causing too much damage to the eukaryotic host, which would be either the person, the plant, or the animal. Other significant organelles found in eukaryotes are the mitochondria and chloroplasts. All eukaryotes have mitochondria. Some of them also have chloroplasts. Things like plants and algae have mitochondria on the top and chloroplasts on the bottom. And things like people, animals, fungi have just mitochondria. Both of these organelles are the site of ATP production. So these are the places where chemical energy is essentially manufactured in the cell. They have double membranes, with the internal membrane being the one that was there before endosymbiosis and the external membrane being the one that developed kind of after endosymbiosis. So when we think back to what I was talking about previously in the lecture, and we mentioned that mitochondria and chloroplasts were the evidence that we have that endosymbiosis occurred, and they're very similar to existing bacteria today. Essentially, mitochondria and chloroplasts were both their own free-living bacteria that got engulfed by another larger cell and turned into, eventually, the structures we know today called mitochondria and chloroplasts. So that inner membrane is basically the membrane that was there when it was a free-living bacterium, and then the outer membrane is the membrane that it acquired over time as a result of endosymbiosis. The inner membrane is where that ATP production actually takes place. So on the mitochondria, which is shown up top, this inner membrane is where ATP is created in eukaryotic cells. And in chloroplasts, these kind of stacks of membrane-bound structures are where the ATP ends up being created. In terms of other significant or important features of the mitochondria and chloroplasts, remember they have their own DNA. So they each have their own little circular chromosome in there. I'm just going to draw it somewhere in here. It's not exactly necessarily where you'd find it in real life, but just to represent it in there. Each of them have their own circular DNA that is separate from the DNA found in the nucleus of the cell that is housing these structures. They tend to divide on their own according to their own cues. That DNA, of course, aside from being circular, which is similar to what we find in prokaryotes, also has a very similar sequence as to what we find in prokaryotes. Also, not shown in here, but you will find 70S ribosomes interspersed throughout the cytoplasm of the mitochondria and chloroplasts. And remember, that is different than what you would find in the cytoplasm of the larger eukaryotic cell. In that place, you would find 80S ribosomes. But in here, inside the mitochondria and chloroplasts, we find 70S ribosomes that are essentially the same ones that we find in bacteria. Another structure we will find in eukaryotes is the cytoskeleton. So just like prokaryotes, eukaryotes also have a cytoskeleton. The cytoskeleton is also made of proteins. They just happen to be different proteins than the ones found in prokaryotes. If you think back to the prokaryote lecture, we talked about those proteins being MREB, FITZ, and CRES. But in eukaryotes, the main cytoskeletal proteins are microfilaments, which are made up of actin, intermediate filaments, which are actually, there are various types of proteins that can make those up. A good example is keratin is a common intermediate filament found in the cytoskeleton of eukaryotic cells. And then microtubules, which are made of tubulin. The function of the cytoskeleton is very similar to what we would find in the prokaryote as well. It helps support the cell, give it some kind of structure. It's involved in cell division, so things like mitosis and meiosis. You probably remember those from introductory biology when the cell is dividing either to make more somatic cells or gametes. And it's also used in transport. Many times when a cell needs to move something around itself on the internal components, what it can do is enclose whatever it needs to move into a small little compartment and kind of attach that to the cytoskeleton and move it along almost as if it were an internal railroad or kind of tram system inside the eukaryotic cell. Another interesting thing about that is prokaryotes, when they are invading eukaryotic cells, may take advantage of this transport system, and they themselves can attach themselves to some of the cytoskeletal proteins and use that to move along until they get to the place they want to go inside the cell. In most eukaryotic cells, we will also find vesicles. Vesicles are typically membrane-enclosed structures. Usually they are temporary in nature, meaning they will kind of come and go as the cell needs them. Typically they're used for things like transport within the cell. They can be used for endocytosis, which means bringing things into the cell. Exocytosis, which means getting rid of things or putting things out of the cell. They can be used for breaking down invaders. So when a prokaryote invades a eukaryotic cell, one of the things that may happen is that prokaryote, that bacteria that's invaded the cell, will get put into a vesicle, something like a peroxisome or a lysosome, and the chemicals that are contained within that vesicle will help break down or destroy that prokaryote that invaded. They can also help deal with waste, storage of nutrients or excess substances, etc. Some specific or important types of vesicles are peroxisomes, lysosomes, transport vesicles, secretory vesicles, and endocytic. Peroxisomes and lysosomes are both involved in degrading substances or breaking them down. So if a eukaryote has been invaded by a prokaryote, that prokaryote might get destroyed by a peroxisome or a lysosome. If there is a big kind of product that the cell has taken in, whether that's a food product or some kind of unnecessary, useless product to the cell that just happens to have gotten inside the cell. A peroxisome or lysosome could be used to help break down that larger structure into smaller structures. Transport vesicles, of course, are involved in movement, whether that is movement around the cell or movement to the external part of the cell. A secretory vesicle is kind of a special vesicle that helps get rid of things like waste products or signaling molecules from the cell. So a secretory vesicle is something that takes molecules and moves them from the inside of the cell to the outside. So secretory vesicles are used to get rid of waste products. They are used to signal to other cells about what is going on inside this particular cell. So let's say you have two cells next to each other, cell A and cell B, not pictured, but we're looking at cell A and we'll say cell B is just right off the screen here. And cell A wants to communicate something to cell B. It might take some proteins that it makes, put it in a transport vesicle. That transport vesicle takes it to the plasma membrane, and then a secretory vesicle grabs those proteins and gets them out of the cell and sends them over to cell B so that cell B can understand the information cell A is trying to communicate with it. And then endocytic vesicles are ones that take substances inside the cell. So these are ones where if a cell has something just on the outside, I'm just going to draw that right here. Let's say something a cell wants to bring inside, whether that is a food particle, whether it's a virus, whether it is a bacterium, whatever. The eukaryotic cell can kind of enclose it with part of the plasma membrane, bring it inside the cytoplasm, and that is an example of an endocytic vesicle. Just like in prokaryotic cells, some eukaryotic cells might have external appendages that they use for movement throughout their environment. In eukaryotes, those types of appendages are either flagella or cilia. So these are considered locomotive structures, or structures that allow them to move. And remember that movement, when we talk about that from the technical word for that in biology, is taxis. And organisms tend to engage in taxis or in movement in response to light or chemicals. So if an organism is moving towards some kind of chemical, that would be what we call positive chemotaxis. If it's moving away from a chemical, that would be negative chemotaxis. If it is moving towards light, it is phototaxis, and that's positive phototaxis. If it's moving away from light, it's negative phototaxis. And so just like in prokaryotes, eukaryotes do these things too. In eukaryotes, those two structures, like I mentioned, that are used for this type of movement are called flagella and cilia. They are structurally identical to each other except for length. Flagella are long and they move like whips, whereas cilia are short and move more like the oars of a boat. So if you can imagine that while you're listening to this lecture or watching this lecture, flagella move back and forth in a whip-like motion, whereas cilia engage in kind of a rowing motion. So when we're comparing the locomotive structures of eukaryotes and prokaryotes, both eukaryotes and prokaryotes have flagella, but only eukaryotes have cilia, and only prokaryotes have pili. So when we look at these structures, they perform similar functions, but they're not identical in terms of exactly the way they move or exactly the structure. The eukaryotic flagella is much more complex than the prokaryotic flagella in terms of the number of pieces that go to make it up and the different types of proteins that are involved. And the cilia are only found in eukaryotes, whereas the pili are only found in prokaryotes. So while they all are involved in movement or taxis, there are some differences between them. Now that we have gone over the basics about eukaryotic cell structure and function, it's time to switch our focus to talking about the 4 types of eukaryotes that we study in microbiology. In our last lecture, we talked a lot about the structures that we find in prokaryotes and the functions of those structures, but we didn't do anything like go over specific types of prokaryotes. And the reason for that is because we're going to spend the rest of the semester basically talking about that. But right now in this lecture, we're going to talk about the four main types of eukaryotic microbes that we study in microbiology, because we don't talk about them much the rest of the semester. So everything in Unit 2, everything in Unit 3, almost everything in Unit 4 will be focused on prokaryotic microbes. And so right now it's time to just go into the eukaryotic microbes a little bit before we spend the rest of the semester talking mostly about prokaryotes. In microbiology, we talk about four main groups of organisms: the fungi, the algae, the protozoa, and the helminths. And just to remind everyone, of course, there are different types of eukaryotes out there. There are people, there are plants, there are animals, there are other types of eukaryotes, basically. But they don't fall into the category of microbiology. So we are not going to talk about those other things. The four types of eukaryotes we're talking about are fungi, algae, protozoa, and helminths, because those are the eukaryotic microbes. The first group that we will talk about or focus on are the fungi. Fungi are significant both medically and environmentally. They are significant medically because they can cause disease in humans, plants, and animals. So obviously when they cause disease in humans, we consider that significant medically. But when they also cause disease in plants, that can interfere with crop production, which means we don't have as much food as a species, which is problematic. And when they infect either our animals, either the ones we keep as pets or the ones that we use as livestock, that can be problematic as well. We can also see the significance of fungi in terms of the environment because they are very, very important in decomposition. We talked about our first lecture about things like nutrient cycling and breaking down dead things so that their nutrients that are in their bodies can be released to other living organisms. Fungi are some of the main things that do that. So they're significant medically because they can cause disease. They are significant environmentally because they're some of the biggest decomposers on the planet. Fungi are also useful to us from an industrial or food production standpoint. Their metabolism and the byproducts that they produce as a result of that metabolism can be very useful in creating foods. Fungi go through a process called fermentation. They can do other types of metabolic processes, but one of their important metabolic processes is fermentation. That fermentation can be useful in making things like bread, things like chocolate, sauerkraut, alcohols. All kinds of different food products are created as a result of this process that certain fungi go through. They can be useful in producing antibiotics. Many of the antibiotics that were discovered originally and many of the ones that we use today are derived from chemical compounds that fungi produce. So these fungi will produce these chemical compounds. We can harvest those, put them in pill form or IV form or injection form, whatever form we want basically. And then we can use those to combat bacterial infections in the human body, the animal body, the plant body, etc. Another thing that fungi are really important with is plant growth. So again, thinking back to our first lecture when I was talking about plants and how the plants need microbes growing near their roots to help them extract nutrients from soils. Those microbes that help them do that are primarily fungi. So in plants that we see growing all over the world, we can look at their root system. And when we look at the root system very carefully under a microscope, oftentimes we will see fungi that are associated with those roots. And those fungi are essentially helping the plant extract nutrients from the soil so that they can be taken up by the plant and the plant is able to grow and photosynthesize and go through its normal life processes. In terms of the structure of fungi, remember fungi are one of the types of eukaryotic microbes that have cell walls. So they have cell walls made of what we call chitin. And fungi can be either single-celled or multi-celled. Fungi that are single-celled are typically referred to as yeasts, and you're looking at some of them up here. Usually they're kind of oval in structure. They can elongate and stretch out and form a structure that we call pseudohyphae, and I'll elaborate on that in just a minute. Multicellular fungi are typically referred to as molds, or oftentimes referred to as molds. They are composed of many cells joined together. They typically are fairly large. A lot of times we can actually see them. They form structures called hyphae, which are just big, elongated masses of the fungus itself that's growing. And when you get enough of those hyphae together, they form what's called a mycelium. So this is an example of a mold, and that mold, when it grows enough and we can see kind of these spindly things sticking out, those individual long projections are called hyphae. And when we get enough of those hyphae together, that's called a mycelium. So going back to the pseudohyphae, the difference between pseudohyphae and hyphae are that hyphae are individual cells that are oftentimes connected to each other by little channels that go through each individual cell. So it's almost like the little tunnels between each individual cells. Whereas pseudohyphae are just individual fungal cells that are attached to each other, but not necessarily connected with that tunnel-like structure. So in hyphae, oftentimes we have individual cells that are connected to each other. I'm going to draw some out in just a second. And their cytoplasms have a way of communicating with each other because of these little spaces. So here's an example of a tunnel. Here's an example of a tunnel. Here's an example of a tunnel. And so basically the cytoplasm of each of these cells can kind of communicate with each other as they're joined together because of these tunnels. Whereas with pseudohyphae, you have individual cells that are joined together and they're connected but not joined by any type of tunnel. They're just individual cells that stretch out and elongate, but they don't have those tunnels between them, connecting them and allowing them to share information. So that's a lot of information, but hopefully that will help put the fungi in context for you in terms of the ones that you have seen in your life and will see in lab. In terms of fungal lifestyle, they primarily are decomposers, which means that they break down dead substances and release the nutrients that are in those substances back into the environment. So when you have a dead tree or a dead animal or a dead plant or even a dead human potentially, the fungi that are in the environment will use that dead body as a source of nutrition. So they will use different enzymes to break down that dead body. They will take the nutrients that they need to grow, and all of the excess nutrients, the ones that they don't need, will get released back into the environment, either the soil, the water, or the air. And then those nutrients become available for other organisms to use and support their own metabolic needs. Some fungi, especially the ones that are very important to us in medical settings, though instead of acting as decomposers, they act as parasites, which mean they actually live on living organisms. So when someone is infected with a fungus, what that essentially means is that fungus is breaking down their body and using their bodily components as a source of nutrition. So the fungi that can act as parasites and can infect people are especially significant to us in microbiology, especially from a medical microbiology perspective, because those are the ones that tend to directly interfere with the structure and functioning of the human body. Our next type of eukaryotic microbe is the group that we call the algae. Now, algae are actually a very, very diverse group of microorganisms. There is one type of algae that technically is a prokaryote, but we group them in with the eukaryotes because most of the algae are eukaryotes. Algae are significant to us in a lot of different ways. In terms of human health, they do produce the majority of oxygen on the planet. So they hang out in the oceans and in other bodies of water, and day in and day out, they go through photosynthesis. They release oxygen. That oxygen ends up in the atmosphere, and they are the ones that produce at least half, if not more than half, of the oxygen that people and animals breathe. They can also be a food source. So there are many cultures around the world that use algae as food sources. Technically, the seaweed that ends up in sushi is a type of algae. So if you eat sushi and there's seaweed that's wrapped around the outer part of that sushi roll, that is a type of algae that you are consuming. They are also significant because they can and do produce certain toxins that can be problematic for people. Sometimes those toxins will get into the respiratory system and cause irritation and cause kind of respiratory distress. Typically, it's not severe enough to kill people, but it can be irritating to the respiratory system and cause trouble breathing, cause just general respiratory issues and symptoms that you would typically associate with having a cold or the flu or something like that. These toxins that algae can produce can also lead to fish kills in certain areas. They can contaminate water sources, and they can just generally cause some environmental problems in certain areas when they get released. In terms of the environment, algae can tell us a lot about the environment, both currently and historically. Algae can be used to tell us whether the water in a particular area is suitable potentially for drinking water. So by looking at the types of algae that we can find in a body of water, we can figure out if that could potentially be a source of good drinking water or irrigation water, things like that. Sometimes algae in the ocean will overgrow and turn the ocean kind of red. This is called a red tide. In conditions like this, oftentimes they're telling us that there is an excess source of nutrients ending up in a body of water, and typically it's not safe for us to be in that type of water. Sometimes the algae that cause red tides can also release toxins into the air and cause respiratory symptoms for certain people, especially sensitive groups. And we can use algae in various industrial applications as well. When we were talking about making biofuels in our first lecture, algae are one of the main sources of producing things like biodiesel and other types of biofuels. So it's possible to take certain types of algae and put them in kind of bad conditions, either a little bit too salty or deprived of certain other types of nutrients, and that helps them accumulate a bunch of lipid inside their bodies, and that lipid can be extracted and turned into fuel. In terms of the structure of algae, they can also be unicellular or multicellular. Typically the ones that are unicellular, like the ones you're looking at here that I'm pointing to, and also the ones here, are referred to as microalgae, and the ones that are multicellular are oftentimes called macroalgae. These will get very, very big. So there are some exceptions to these rules, but these are just kind of general rules here. Seaweed is an example of macroalgae, and you guys are all probably familiar with that. You can see that this type of algae gets very, very big, and it has trillions of cells that make up its body. Because the algae are such a diverse group of organisms, there is no one single molecule that makes up the cell walls found in these organisms. There are lots of them. I've got some of the molecules written out here. Cellulose is one of the most common structures or most common molecules that makes up the cell wall in algae. Cellulose is a bunch of individual glucose units strung together, so it's a polysaccharide, and it is the same type of molecule that makes up the cell wall in certain plants. Calcium carbonate is another type of molecule that can be found in the cell walls of certain algae. It's kind of a hardened substance. It can be used to make things like chalk, and if you've ever heard of the White Cliffs of Dover, they're basically cliffs that you find in England, and they're pure white in many of the places, and it's because a bunch of the skeletons of the algae made of calcium carbonate have accumulated over the years and kind of built up. So you can just see these cliffs that are just white because of the skeletons of algae. And then silicon dioxide is another common one. Silicon dioxide is another term for what is essentially glass. And so there's a certain group of algae called the diatoms. That have cell walls that are basically glass. And you're looking at some of the diatoms right here. Diatoms are they're a very diverse subgroup of algae, and they have a lot of different shapes. Our third type of eukaryotic microbes are the protozoa. Protozoa are animal-like organisms. They are always single-celled. They don't have cell walls, and their internal structure is very similar to what we would find in animal cells. In fact, their name, protozoa, actually breaks down to meaning first or before animal. So they are not technically animals, keep that in mind. They are just very similar to animals. They are kind of like the precursor to animals, or what came before animals. They are also another fairly diverse group of microbes, and they have some pretty significant environmental impacts as well as impacts on humans. We'll get into those impacts on the next slide, but first, just talking about their general structure. Like I mentioned before, they are single-celled, they don't have a cell wall, and internally they are very similar in terms of what we find in animal cells as well. So the structures that we find in animal cells, which are the ones that you've probably talked about in previous classes, your general biology class, and if you've taken anatomy and physiology, you've talked about human cells in depth. And so the protozoa are similar in nature to those types of cells. There are 4 main groups of protozoa, basically based on their locomotive structure or their means of getting around. The first group is called the flagellates. And the flagellates move by flagella. You're looking at some flagellates up here, just at one of the examples of flagellates that we have here. And you can see the flagella coming out of their cell structure right there. So these ones move around by flagella. The next group of protozoa are called the ciliates. And the ciliates move around by cilia. So sometimes the cilia are a little bit harder to see because oftentimes, instead of being very large, obvious structures like we see in the flagellates, they will be kind of just located in maybe one area on the organism. I'm just going to circle where they are here. So in this case, right in this area, this kind of groove area, that's where we actually find the cilia for this particular organism. And you can see some of them here as well. If you have the PowerPoint or the PDF of these slides open on a screen, whether it's a laptop or iPad or other kind of type of tablet or phone, and you zoom in, you'll be able to see those cilia that are protruding from that groove area on that microbe. The next group of protozoa is called the amoebas or the pseudopodia. These move around by what we refer to as a pseudopod, which means false foot. And essentially what these ones do is when they want to go somewhere, let's say they recognize that there's a chemical in their environment that they want to take in and consume as food, they will take the cytoplasm that is in their cell and kind of shove it around to one area of the cell. So they will take the cytoplasm from maybe the bottom part of their cell, and they'll shove that cytoplasm into what we would consider, looking at right here, the top part. and that will drag the rest of the body of this organism along with it. So that's how they move. They move in this kind of, I would say it's kind of like a crawling type motion. It's a little bit smoother than crawling because really it's just cytoplasm being shoved over and over so that the body is drug along with it. And then the last group of protozoa are called the apicomplexan. And these ones actually don't move on their own. These ones don't have a locomotive structure, and as a result, they rely on a host to move them. So one example of these types of organisms are the protozoa that cause malaria. Malaria is a disease caused by protozoa. It's found primarily in the tropical regions of the world, and it is spread by mosquitoes. So if a mosquito bites someone who is infected with this protozoa and takes in some of their blood, and that blood happens to have the protozoa in it, and then that mosquito goes and bites someone else, a little bit of that blood from person A is transferred to person B, and in many cases, the protozoa from person A will then also be transferred to person B. So this type of protozoa relies on a vector or a host to move it around. In this picture, the little pink dots that you can see in the larger pink cells are the protozoa. So they are actually living inside these blood cells, and that's how they survive when they're in the human body. One final thing about basic protozoa structure is that some protozoa can form what we call cysts. Not all protozoa can do this, but some protozoa form a protective structure around themselves called a cyst when environmental conditions are not very great. So if it's too cold, or maybe they are getting dehydrated, or there's not enough nutrients, they can kind of put themselves into this cyst and go into a brief pause so that they're protected during these difficult environmental conditions. And then when environmental conditions get better, they can come out of their cyst and turn back into a normal metabolizing protozoa. In terms of the environmental significance of protozoa, many times protozoa serve as some type of predator to other types of microbes, especially things like algae, bacteria, and fungi. So oftentimes protozoa live out in the environment in some kind of body of water, like a lake, a river, or a stream. They might even live in the soil sometimes. And one of their main sources of food, in many cases, is some other type of microbe, like an algae, a bacteria, or a fungus. As a result, these protozoa are able to help keep populations of these other types of microbes in check so they don't overgrow. Some protozoa can also be used in industrial applications. One example of this is that certain species can actually be used as a type of insecticide. What happens in this case is the protozoa are grown in a laboratory setting, and they are put into kind of a difficult environmental condition so that they form a cyst. Once the cyst is formed, we can extract the protozoa cysts from the laboratory setting, put them into some type of solution, and then spray them on crops. This particular type of protozoa that is used is only infectious to things like grasshoppers and fire ants, and this allows us to prevent crops from being damaged by those types of insects without having to use some type of chemical that might be damaging to the human body. So basically, even though these protozoa get put on the food, since they are not infectious to humans, it doesn't really matter that they're there. Once we process the food, we clean the food, most of them are gone, and even any remaining ones, they aren't infectious to humans, so they just pass through us. Another significant aspect in terms of environmental factors for protozoa or environmental impacts for protozoa is in animal digestion. We talked about this a little bit in the first lecture, But protozoa are often involved in helping certain types of organisms break down the food that they consume so they can actually get to the nutrients. One example of this is termites. So termites are insects that consume wood. Wood is basically their food source. But the termites themselves cannot actually break down the wood. Without help from the protozoa that live in their guts, the wood would be useless to the termites. But because of the protozoa that live inside the GI tract of these insects, the termite is actually able to get energy from the wood source. So these protozoa are significant in terms of helping certain organisms break down food from their environment. And then for human health, protozoa in general can be a significant source of disease. I've listed a couple here. We'll get more into these later in the disease unit. But malaria, that I mentioned on the previous slide, is a mosquito-borne disease that causes problems for humans primarily in the tropics, but all over the world. So globally, malaria is a very significant cause of morbidity and mortality. We don't see a lot of it in the United States because most of the United States is not in the region where the mosquito that transmits this protozoa is found. So we typically don't see a lot of that in the US, but all across the tropical regions of the world, there are people who are sick and dying of this disease every day. Another disease that's fairly significant that a lot of people don't know about or haven't really heard about is one called trichomonas. This is actually a sexually transmitted infection that's caused by a protozoa. So oftentimes when people think about sexually transmitted infections, they think about things like bacteria and viruses, and many of the STIs that are out there are bacteria and viruses, but there are protozoa that can cause sexually transmitted infections as well. And then just a final example that people may have heard of before is Giardia. Giardia is caused by a protozoa, and when people ingest this protozoa into their GI tract, they often end up with yellow, foamy diarrhea for several weeks. Some people get over it on their own. Other people need the help of medications and supportive care to deal with the infection. So protozoa do have a significant impact on human health, mostly in kind of a negative way in terms of once they get in our body, they cause problems, but they do have positive environmental and industrial uses as well. Our final group of eukaryotic microbes are the helminths, which are the infectious worms. Helminths are animals. While protozoa are similar to animals, they're not technically animals, but helminths do fall into the kingdom of Animalia, so they are animals. They are always multicellular. They do not have cell walls, and they are another fairly diverse group of organisms. Obviously, there are a lot of worms in the world, but not all of them are ones that we study in microbiology because they don't infect humans or animals. But the ones that do infect humans and animals are generally lumped into the study of microbiology. They're put into this category because even though as adults they can get rather large in some cases, as they reproduce, their reproductive structures are very, very tiny and are grouped into microbiology, oftentimes at least partly as a result of that. Most helminths have very complex life cycles. Oftentimes they need multiple hosts to live in before they become infectious to people or other types of animals. They typically have very specialized bodies. They are highly evolved parasites, meaning they're very, very good at their job of being parasitic. Sometimes they lack a digestive system. The reason for that is they don't necessarily need a digestive system because they are just going to steal nutrients from their host or hosts, and they're going to rely on the host to do or have done most of the digesting for them. So they don't need a digestive system because they're just going to steal nutrients from their hosts. They may lack some type of locomotive means, meaning they don't move on their own, because again, they're just going to rely on their host to do that. They have evolved so well that they have essentially figured out how to get the host to do all the work for them. And the thing that helminths tend to be very, very good at and have a very highly developed system for is rapid reproduction. Many, not all, but many helminths are able to produce something like 20,000, 30,000, 50,000 eggs per day. And some of them are not much more than a head and just a bunch of digestive structures. The one I'm pointing out right now in the lower left-hand corner is an example of a tapeworm. And a tapeworm basically has a head, which is also called a scolex. And everything else, all of these other segments you see here, are primarily used for reproduction. So a tapeworm is a head and a bunch of reproductive structures for the most part. So helminths are very, very good at reproducing quickly and in high numbers. There are two phyla of helminths that we pay attention to. There are what we call the platyhelminths, which are flatworms. And within the flatworms, there are two classes that we pay attention to. The trematodes, which include the flukes. This is an example of a fluke. And the cestodes, which are the tapeworms. And this is an example of a tapeworm. We will talk more about tapeworms and flukes in the disease unit. Right now, just focus on learning that vocabulary, and then we'll get into the structure, the function, and the diseases they cause later. The other phylum that we pay attention to in microbiology are the nematodes. These are roundworms. There's an example of one right here. And just a common example of a roundworm that you may have heard of are pinworms. It's fairly common for especially children all over the world to acquire this infection, and you will notice it because you may see little tiny white worms that are wiggling around in fecal matter. So sometimes it's very obvious when a child has pinworm infections. But that's just one example. There are other types of roundworms as well. Pinworms are just some of the most common and ones that many people get at least once in their life. Now, helminths in general are not super common in the United States. Most of our sanitation and our behavioral practices surrounding food, waste removal, disposal, etc. help stop helminth transmission fairly well. But in most of, or maybe not most, but in much of the rest of the world, helminths can be a significant problem. In places where there is not necessarily a great sanitation system, where human waste is not dealt with in the ways that we know that it needs to be dealt with to fully make sure it's not getting back into our food system or our water system or onto crops and things like that, helminth infections are fairly common. Also, in certain places where people live a lot closer to nature, maybe they come in contact with soil a lot, they come in contact with livestock a lot more often, helminth infections can be common in those areas as well, no matter what the sanitation system is like, because we can typically find certain types of helminths in those conditions as well. So while helminth infections are not super common in the United States, aside from maybe pinworms, many people across the globe will develop helminth infections. Oftentimes they are infected with more than one helminth at a time. And for some people, basically they acquire infections as young children and are infected with helminths the rest of their life. So for lots of people across the globe, they have worm infections from shortly after the time they are born until they die. In terms of complications of infections with helminths, if a helminth infection is treated fairly quickly, and there are good medications to do that in many cases, but if it's treated fairly quickly, usually there aren't a lot of long-standing consequences for the person. However, if we're talking about a lung infection or repeated infections, there are potentially several consequences. One is internal bleeding that leads to anemia. One of the reasons that this may happen is that helminths oftentimes have some kind of special structure, like a hook or a barb, that allows them to dig into our tissue and stick there. And as a result of digging into that tissue, we start to bleed a little bit. As the helminth reproduces and maybe makes more helminths, then more and more helminths get stuck in maybe the GI tract or another type of organ or something, and more bleeding occurs. Not to mention then our body's trying to fight off this infection and trying to get rid of this organism, and the immune cells that are involved in that can cause additional tissue damage and lead to additional bleeding. So for people who have severe worm infections, anemia may be one of the results. Other types of issues that can occur with helminthic infections are nutrient deficiencies, especially in children. These occur for kind of two main reasons. One, the helminth uses the nutrients from the host, or from the child in this case. And in countries where children have access to a lot of food and nutritious food, that may not make too much of a difference. But if you're in a situation where a child only has one meal a day, or they're only getting one source of food, maybe they're only getting rice, or they're only getting eggs, or they're only getting some type of fruit or vegetable, and not much else during their day-to-day activities, then that can be especially problematic because the little nutrients the child is getting are being absorbed by the helminth. Additionally, again, coming back to these special structures like the hooks and the barbs that the helminth oftentimes uses to stick in the body, that can lead to scarring, especially in the GI tract. So the scarring will end up interfering with nutrient absorption. So the nutrition that the child is getting is not getting absorbed by them. The helminth gets potentially even more of it, and so the child is deprived of even more nutrients. And then kind of related to internal bleeding, nutrient deficiencies, everything else, just the general organ and tissue destruction that occurs when a helminth invades the tissue or the organ causes problems for the person. If you think about the liver being invaded by a bunch of different worms, or the GI tract being invaded by a bunch of different worms, those organisms take up space, they cause damage, they steal nutrients, etc. And they basically just interfere with the proper functioning of the organ or tissue or whatever structure the body is supposed to perform. So helminths generally are not a positive thing. There are some situations where they might be able to be beneficial for us, but that needs a lot more research. And in general, helminths, not necessarily the most pleasant type of microbe to deal with.